Hello everyone, my name is River Medlin and today I'm going to be talking about oxidative stress and its potential as a bactericidal agent within bacterial infections. Just kind of an overview on antibiotics. It is a class of drugs that are used to treat bacterial infection under the umbrella term of antibiotics. There are several different classes. Each class contributing to a different mechanism to target specific bacterial infections. The improper use of those drugs, however, can lead to antibiotic resistance. Improper use, such as not taking when directed or as directed by your doctor, can lead to organisms developing resistance to those drugs, which make them harder to treat. Antibiotics under that umbrella term can be broken into bactericidal or bacteriostatic drugs. Bactericidal drugs, some of the examples are aminoglycosides, beta-lactams, quinolones, and polymyxins. Bacteriostatic drugs include macrolides, sulfonamides, and tetracyclines. Those are not too important for this presentation, but just kind of an overview just to introduce you to some terms within the antibiotic drug category. My focus deals with oxidative stress. Oxidative stress is a physiological response within a cell caused by reactive oxygen species. Reactive oxygen species are harmful chemicals that are very reactive to different intracellular components. In that example there, you see a normal cell encountering things that we call free radicals. Free radicals are a form of reactive oxygen species in which the reactive oxygen species interacts with the cell, causing the cell to break apart and die. A few examples of reactive oxygen species include superoxides, hydrogen peroxides, and hydroxyl radicals. Superoxides are a very dangerous form of gaseous oxygen where there's an extra electron found on the diatomic molecule. Superoxides are known to be broken down through a normal process within a cell known as the Fenton reaction. Within the Fenton reaction, the superoxide is broken down into the hydrogen peroxide, which is a less harmful form than the superoxide. The cell does this through a specific enzyme known as superoxide dismutase. The superoxide dismutase enzyme turns the superoxides into hydrogen peroxide, at which point the hydrogen peroxide is less toxic for the cell. However, in some instances, we do see the hydroxyl radicals being formed through the Fenton reaction whenever intracellular iron is present, at which point the hydroxyl radicals are much more dangerous than the hydrogen peroxides. The hydroxyl radicals interact with cells, and we're going to get more into that here in a minute, but the hydroxyl radicals are much more dangerous for the cells than hydrogen peroxide is. I do want to make it clear, though, that all three of these examples are very toxic to cells. Like I said, we will get into the different effects of this oxidative stress mechanism. The first example would be lipid peroxidation. Lipid peroxidation is an interaction between free radicals and intracellular membranes. You might have heard of cell membranes or plasma membranes or membranes of organelles. When free radicals and reactive oxygen species are present, those membranes can interact with those molecules, causing the membranes to break apart or be destroyed, in which case the integrity of the cell has been compromised and the cell can no longer perform its normal functions. The next example is protein carbonylation. This is a biochemical process in which the oxidative stress causes normally folded proteins to lose their functional shape and turned into a less functional form where the proteins can no longer perform their normal function, causing a failure to perform cellular functions, in which case the cell can no longer perform the necessary processes for cell life. The third example is DNA damage, when free radicals interact with intracellular DNA. Things known as double-strand breaks and single-strand breaks can occur, where the DNA molecules actually break apart and the cell can no longer perform the necessary functions that DNA is intended to do. And the cell can no longer read, transcribe, or translate the genetic material. We see changes in gene expression and a failure to perform chemical processes, causing problems with cell life and cellular replication. My question was, can oxidative stress be used to treat bacterial infection? I performed a meta-analysis of 16 different articles looking at the presence of reactive oxygen species when treated with bacterial antibiotics. The second aspect that I looked at was the bacterial lethality, meaning what is the correlation between reactive oxygen species found within the cells and the cell death being observed. And so I'm not going to go too much in depth on this table, but I do want you to notice that in most studies, the presence of reactive oxygen species is observed, denoted with a number 1, and only one study showed that no reactive oxygen species was observed. The other category that I looked at, bacterial lethality, most of the studies I found was dependent on what antibiotic was used and what organism was the observed organism correlated with the study. There were a few studies, as you see, denoted with a number 1, where reactive oxygen species and bacterial lethality were correlated with the antibiotic and the intended organism. And so those were considered successful studies. And there was only one study where there was no correlation between a presence of reactive oxygen species and a cell death scenario. All right, here's a graph that I created, just kind of summarizing some of the information that I found. Approximately 81% of the studies showed presence of reactive oxygen species in the model organism. It's a pretty substantial portion. None of my results were statistically significant. However, more needs to be researched to provide a more substantial sample size, in which case more conclusions can be drawn from the data that is observed. Like I said, there were 2 categories I looked at, the second being lethality of reactive oxygen species. Approximately 50% of the studies that I observed induced cytotoxic effects within bacterial organisms. Approximately 31 was dependent on which organism was being studied versus which drug was being used on that organism. And then approximately 13% of the studies did not show any statistically relevant data. My research focused on using a chemical to treat bacterial infection. So my proposal is that since this pathway has been observed in several different antibiotic versus bacteria scenarios as a potential primary pathway, I'm proposing that this secondary pathway known as oxidative stress can possibly be exploited to treat antibiotic-resistant bacteria. If this is a potential therapeutic option, this would open the door for more antibiotic development, I know right now there's a lot of drug development with the newly developed coronavirus, COVID-19. And so all of this is very relevant right now in the world. I hope that this study gives you a little insight into antibiotics and antibiotic development and bacterial infection. But hopefully, if this is a potential option for bacterial infectious disease, then hopefully we'll see a better outlook for life-threatening infections like MRSA and VRSA and multiple pan-resistant bacteria. So there's a few of my images and references. As always, if you have any questions, feel free to let me know. Feel free to shoot me an email. Thank you for your time. And I'd like to give a special shout out to Dr. Rose and Professor Duke for their help with this research. Thank you, and enjoy the rest of SLAM.